Hi students, in this lecture video I'm going to be talking to you about anatomy and physiology of the ear and hearing. Okay, so let's look at kind of a general schematic of the ear and how it is divided according to anatomic landmarks. So essentially we have three sections of the ear. We have our external ear, which is comprised of the fleshy outer part of the ear that's known as the auricle or pinna. These are interchangeable terms, so either one you want to use is fine. It's basically just your personal preference and what you can remember better. So this whole outer part of the ear is your auricle or your pinna. The helix is the most lateral rim of that outer ear, and then the lobule is the earlobe itself. Now the boundary between the external ear and the middle ear is this structure known as the tympanic membrane. That's colloquially referred to as the eardrum. So for the purposes of an anatomy class, you should refer to it as the tympanic membrane, but just know that eardrum is basically the common term that's used for that structure. So external ear, medially, it's going to be bounded by that tympanic membrane, and then the external ear, of course, just ends at that helix, which is that outer rim of the auricle or pinna. The external acoustic meatus, now it's what you might see some discrepancy of depending on which author you're referring to. Saladin treats the external acoustic meatus as being basically the same thing or part of the ear canal. The way I learned this was the external acoustic meatus is restricted to the opening in the temporal bone. So this would be the outline of the temporal bone here. The opening is actually the external acoustic meatus. So I believe most authors treat the external acoustic meatus separate from the ear canal. The ear canal itself would just be this fleshy tube. So that portion of the external ear that abuts the tympanic membrane. So that is the external ear. The middle ear is from the tympanic membrane to the structures that are kind of known as the bony wall, basically at the oval window here. This is the stapes, one of the bones of the middle ear, and that abuts a structure called the oval window. And then inferior to that is a structure called the round window. So basically this fairly small area between the tympanic membrane and the round and oval windows is going to be your middle ear. So we have our tympanic membrane and then the three middle ear bones, the malleus, the incus, and the stapes. And then your internal ear, sometimes this is just referred to as the labyrinth. The internal ear is going to hold your organs of hearing and your organs for equilibrium. So the cochlea contains the organ of hearing, and the vestibule and semicircular canals are basically for equilibrium. And then what we see here are the two nerves that will send the signal from the organ of hearing and the organ of equilibrium, and eventually those two nerves are going to join together into the vestibulocochlear nerve, and that is a cranial nerve. So that is the basic overview of the three regions of the ear. So I'm going to be talking to you a little bit more about what the functions of each of these parts are, and also giving you more details about how we actually are able to hear. So really the external ear and the middle ear are what are involved with hearing, whereas the internal ear, which is the labyrinth, functions in both hearing and equilibrium. And our receptors for hearing and balance are separate sensory receptors. So they are responding to different stimuli, and they are activated independently. But as I said previously, the vestibular nerve and the cochlear nerve will eventually join together into cranial nerve number 8, which is the vestibulocochlear nerve. So cranial nerve number 8, otherwise known as the vestibulocochlear nerve, carries both signals from equilibrium and hearing into the brain. Okay, so our external ear, we have that auricle or pinna. That's going to be composed of the outer rim, which is known as the helix, as well as the lobule or the earlobe. And then the external acoustic meatus, otherwise known as the auditory canal. Like I said previously, Saladin treats this as basically one and the same with the ear canal. I like to make the distinction between what I consider the external acoustic meatus proper and then the flesh-lined tube that reaches all the way to the tympanic membrane, which is generally referred to as the ear canal. But this is a short, curved tube that's lined with skin. Of course, it's going to bear some hairs as well as sebaceous glands and ceruminous glands. Those are the glands that secrete earwax. So earwax is going to be secreted by those ceruminous glands. Another term for earwax is cerumen. And then also considered part of the external ear is that tympanic membrane. And really the tympanic membrane is the barrier between the external and the middle ear. Again, colloquially, this is referred to as the eardrum, but for the purposes of your anatomy and physiology class, you should refer to it by its proper name, the tympanic membrane. It is the boundary, like I said, of the external and middle ears. It's basically just a thin, sort of drum-like structure that is comprised of connective tissue. It has the appearance of basically being stretched taut between the superior part of the ear canal and the inferior part of the ear canal. And what it does is it vibrates in response to sound, and it transfers the energy of that vibration to the bones of the middle ear. And we'll see how that works momentarily. So the middle ear is a small air-filled mucosa-lined cavity within the temporal bone. Laterally, this is going to be flanked by the eardrum. Medially, it's going to be flanked by the bony wall that contains the oval or vestibular window, as well as the round or cochlear windows. And another structure that's considered part of the middle ear, even though it proceeds medially and at some parts is essentially lined up with the inner ear, is the eustachian or auditory tube. So the eustachian tube or auditory tube, again, you can use either of these terms interchangeably. It's going to connect the middle ear to the structure known as the nasopharynx, which I will show you a diagram of momentarily so you get an idea of how this is connected with the anatomy of the head and the throat. What this does is it equalizes air pressure within the middle ear cavity with the external air pressure. So if we take a look at the nasopharynx, nasopharynx is essentially this region that is deep to the nasal cavity. So the nose, of course, it's basically at the very posterior portion of that nasal cavity, and it's going to be continuous with the region known as the oropharynx, and then the hypopharynx below that. So this whole structure here is the pharynx, and of course this has numerous functions in terms of air intake, connecting the oral cavity to the esophagus and the nasal cavity to the trachea. So let's take a look at the middle and internal ears. This would be basically showing the cut temporal bone. Here's your tympanic membrane, which is going to be that boundary between your external ear this way and your middle ear this way. We have 3 auditory ossicles or ear bones, which I will talk about momentarily. And then we have the oval window, which is deep to the stapes. So the stapes is going to abut this oval window. And then we also have the round window. And essentially where the round window and the oval window line up is the boundary of that middle ear. And then of course the eustachian or auditory tube is going to proceed medially and inferiorly. So the ear ossicles are the small bones within the tympanic cavity. And these are named the malleus, the incus, and the stapes. So malleus basically means hammer, incus, anvil, stapes, stirrup. And oftentimes those terms are basically used interchangeably, hammer, anvil, and stirrup. But again, those are generally more colloquial terms. So the ear ossicles are suspended by ligaments and joined by synovial joints. They transmit vibratory motion of the eardrum to the oval window. And then we have a couple of protective mechanisms that actually help to protect our hair cells within the ear from loud noises. So we have a feedback system that involves a number of different muscles, including the tensor tympani and the stapedius muscles. And those are going to contract reflexively in response to loud sounds in order to prevent damage to the hearing receptors. Now, what's interesting about that is they apparently evolved in response to loud, slowly building natural sounds, thunder, for example. But the lag time for the contraction of those muscles is too long for that to be a protective mechanism from very loud synthetic, artificial, human-caused sounds, such as gunshots or fireworks. And they don't serve much of a protective function for sustained loud noises, such as the sound of machinery, for example. So there are built-in mechanisms that essentially help to dampen sound within the ear. But again, those are not necessarily going to be protective in all situations. Okay, so if we look at the middle ear from this viewpoint here, we have our eustachian tube. We have a temporal tympani muscle, which actually helps to kind of brace the tympanic membrane and allow for less sound to be transmitted from the external ear into the middle ear. And then we have the malleus being the most lateral bone, the incus being the middle bone, and then the stapes being the most medial bone. All right, so our internal ear, this is often referred to as the bony labyrinth. It contains complex channels within the temporal bone that are lined by a membranous labyrinth, which is filled with a fluid known as endolymph. This is going to be similar in composition to intracellular fluid, or ICF, but it's going to be particularly rich in potassium. And then we have perilymph between the bony and membranous labyrinths, which is similar in composition to cerebrospinal fluid. Now the various chambers within this bony labyrinth that are filled with fluid are going to play a very important role in transmitting sounds that are funneled into the ear. And that's going to be essential for the process of sending action potentials into that vestibulocochlear nerve. Now, the vestibule is a central egg-shaped cavity within the bony labyrinth, and this is really an organ of equilibrium rather than hearing. So it contains two membranous sacs, the saccule, which is continuous with the cochlear duct, and then the utricle, which is continuous with the semicircular canals. And these sacs are going to house equilibrium receptor regions known as maculae that respond to gravity as well as changes in the position of the head. So again, this largely has to do with equilibrium rather than hearing. At the very end of this lecture, I'll talk briefly about equilibrium and how we're able to sense position in space. All right, so let's take a look at the temporal bone with how all of these components fit in. So essentially what this is showing is the utricle and saccule are in this general region, semicircular canals, which are part of this system for equilibrium, and then the cochlea is somewhat more anterior. So nerves from those two structures are going to join together to form that vestibulocochlear nerve, or cranial nerve number 8. Now, if we were to look at these two systems in isolation out of the skull, this is what it would look like. Again, we have the saccule and the utricle, and the semicircular canals, which are involved with equilibrium. And then the organ of hearing is actually contained in this structure that kind of resembles a snail shell. That is the cochlea. So this would be the cochlear nerve here, the vestibular nerve here, where they join together, vestibulocochlear nerve. And then looking at it from a slightly different view here, we have our tympanic membrane, so this would be most lateral, malleus, incus, and stapes, abutting the oval window. That's ultimately going to allow for vibration to have an influence within these fluid-filled cavities, and that's largely how we are able to hear, which I'll go into some detail about the mechanism of actually generating action potentials via depolarization of hair cells in a moment. Okay, so the cochlea is a spiral conical bony chamber. It extends from the vestibule and it coils around a bony axis known as the modiolus. So superficially, the cochlea really does bear a strong resemblance to a snail shell. The cavity of the cochlea is divided into 3 fluid-filled chambers. The scala vestibuli, which abuts the oval window and contains that fluid known as perilymph. The scala media, or the cochlear duct, which contains that fluid known as the endolymph. That's the fluid that's really rich in potassium. And then within that cochlear duct is the organ of hearing itself, which is known as the spiral or acoustic organ. And then the scala tympani terminates at the round window and also contains perilymph. So essentially we have this sort of sandwich-like effect with the scala vestibuli and the scala tympani sandwiching this scala media with the actual organ of hearing in the middle region. The spiral organ converts vibrations into nerve impulses, and the epithelium of the spiral organ is comprised of hair cells and supporting cells. Now, these, of course, are not literally hairs. They're called hair cells because they bear a superficial resemblance to hairs, but the hair cells essentially have a cell body and then a structure known as the stereocilia. And there are many different hair cells within these structures, and they are basically lined up in rows with the stereocilia extending into this fluid. So there's 2 different types of hair cells. There's the inner hair cells and the outer hair cells. The inner hair cells are really more involved with the majority of our ability to hear, whereas the outer hair cells serve more of a purpose in adjusting the response to different frequencies, and they help to enable the inner hair cells to work with greater precision. So those stereocilia on the hair cells, again, they bear a superficial resemblance to hairs and also cilia, stereocilia. They're not technically cilia based on the internal structure, but they are going to be these kind of long filamentous processes that come out of those hair cell bodies. Okay, so various different views of the cochlea. Cochlea itself in this topmost image here, you can see that if we were to take a cross section of this, each of the coils is going to have this same pattern with the scala vestibuli up top, the the scala media in the middle, and then the scala tympani on the bottom. So it's basically just going to be this repeating pattern. And it's the middle section that contains the endolymph that actually has the organ of hearing in it. So the cochlear duct, or the middle part of this structure here, contains that potassium-rich endolymph, and then the hair cells themselves are in that middle region. So if we were to look at that up close, we would see our individual hair cells here. So the hair cells are in purple. We have supporting cells shown in pink. And then the stereocilia are those tall cilia-like projections that extend into this fluid known as the endolymph. And there's a couple different anatomical landmarks that you should be aware of here. We have the basilar membrane, which is below the hair cells, and then the tectorial membrane, which is above the hair cells. And that's going to be important in terms of when vibration does get transmitted to this area, the ability of the hair cells to generate to generate an action potential to respond to what we're hearing is going to be based on the movement of that fluid and the movement of the tectorial membrane. So what happens when we have air vibrating is it gets funneled into our ear via the ear canal. It's going to come into contact with our tympanic membrane. That tympanic membrane is going to oscillate when vibrating air reaches that, and it's going to transmit those vibrations into that system of inner ear ossicles, the ear bones. So essentially we have that malleus, incus, and stapes, which are going to be absorbing that vibrational energy from the tympanic membrane, transmitting that energy into the fluid of the cochlea, and hence the organ of hearing. So what we see within that organ of hearing is that the inner ear fluid is going to move. So the inner ear fluid as well as the basilar membrane are going to move the hair cells, and that causes depolarization from an inward flux of that potassium-rich endolymph. Now, we all know from looking at how action potentials are generated that in order to send an action potential, you need to have depolarization of a neuron. Depolarization is going to depend on an inward flux of an ion that makes the intracellular fluid more positive. And in most contexts that we've looked at up to this point, we've seen an inward flux of sodium. But both sodium and potassium are positively charged ions. So in this case, we see an influx of potassium. Again, that's a positively charged ion. It's going to serve the same general purpose to depolarize the cell. And as a result, a neurotransmitter is released from the base of the hair cell, and the signal within the cochlear nerve is generated. Now, your book goes into a lot more detail with how this phenomenon works with structures known as tip links, which are protein fibers that extend between one stereocilia and another. So essentially what you get is the movement of this basilar membrane that causes the hair cells to bend. And when that bending occurs, these tip links pull on these proteins, these gates, at the tips of those stereocilia. It pulls those open and allows for an inward flux of that endolymph, thus allowing for depolarization of the cell to occur. But basically, the level of detail that I have communicated with you is kind of the level of detail that I would like you to know going forward. All right, so I did want to briefly mention the fact that we have this mechanism called cochlear tuning. I'm not going to go into a lot of detail with this, but if you are interested in learning more about sort of the fine-tuned responses to different types of sound, I do encourage you to read this section in your book. But cochlear tuning is essentially a way that our outer hair cells will kind of mediate our response to different pitches and frequencies. And then another sort of feedback system that we have in place is the tympanic reflex, which help to essentially, it helps to dampen the amount of energy that's ultimately transferred into the middle and inner ear. So that is part of that protective mechanism that is in response to loud noises. And then I did just want to point out the general pathway that we see here with these neurons. We have the cochlea here, of course, and the vestibule here, which this is primarily going to be used for equilibrium, and the cochlea is primarily used for hearing. So the nerve fibers, or the axons of the neurons involved in this, are ultimately going to join with one tract, so cranial nerve number 8, the vestibulocochlear nerve. And it's going to synapse in this region of the pons very close to the medulla. There is a very short interneuron here that is going to synapse with a neuron that ends in the inferior colliculus. And then we're going to see another neuron that proceeds up to the thalamus, and finally a neuron that proceeds into the primary auditory cortex. So that's part of the cerebral cortex. Okay, so just as an overview, this is kind of a nice sort of simplified diagram that goes through these major stages of hearing. This is tracing the route of sound waves through the ear. So we have sound waves that vibrate the tympanic membrane. Again, the tympanic membrane is this sort of drum-like membrane that separates the external ear from the middle ear. The middle ear is going to be this region that contains the beginning of the eustachian tube up until this bony wall with the oval window, which the stapes is abutting, as well as the round window. That vibrational energy is essentially going to be transferred through these ossicles. So the auditory ossicles are going to vibrate, the pressure is amplified through this system, and then the pressure waves that are created by the stapes pushing against this oval window move fluid within the scala vestibuli. So this is what we're seeing here, the fluid basically being displaced. And then we have sounds with frequencies below hearing travel through a part of the structure known as the helicotrema. They don't actually excite hair cells because it's not something that we are tuned to respond to. And then we have sounds within our hearing range that go through the cochlear duct, vibrating that basilar membrane and deflecting those hairs on the inner hair cells. And again, that's essentially going to cause depolarization to occur such that we can generate an action potential to send through cranial nerve number 8. And then the last thing that I just wanted to mention is equilibrium. So equilibrium, separate sensory system from hearing, but they're located very close to one another. So the vestibular apparatus houses the receptors for equilibrium. We have the semicircular ducts, also known as the semicircular canals, which respond to angular acceleration and change in the rate of rotation. And then we have the saccule and utricle, which are used for static equilibrium, perception of head orientation, and linear acceleration, as in, for example, when we're just moving forward in a car. So those will be able to sense a change in velocity in a straight line. All right, so please let me know if you have any questions about this material. And in class, we'll be reviewing parts of the ear with an examination of ear models. Study wisely. As always, let me know if you have any questions.